Hello, everyone. Welcome back to Meds.Concepts. In the previous lecture, we studied transcription in prokaryotes. Today, we will explore how transcription occurs in eukaryotic cells, which is significantly more complex due to chromatin organization and the involvement of multiple RNA polymerase and transcription factors. In this lecture, we will discuss RNA Polymerase I, II and III, promoter architecture, general transcription factors, pre-initiation complex formation, CTD phosphorylation, elongation factors, chromatin remodeling, and transcription termination. Features of eukaryotic transcription. Eukaryotic transcription differs considerably from prokaryotic transcription. First, transcription occurs inside the nucleus, whereas translation occurs in the cytoplasm. Therefore, the two processes are spatially separated. Second, eukaryotes possess multiple RNA polymerases, each responsible for transcribing specific classes of genes. Third, DNA is packaged into chromatin, creating a physical barrier that transcription machinery must overcome. Fourth, transcription requires a large number of accessory proteins called transcription factors. Finally, RNA processing events such as capping, splicing, and polydenylation are closely linked to transcription itself. Therefore, eukaryotic transcription is highly regulated and significantly more complex than bacterial transcription. Eukaryotic RNA polymerase. Unlike prokaryotes, which poses a single RNA polymerase, eukaryotes contain three major nuclear RNA polymerases. RNA polymerase I synthesizes the large ribosomal RNA precursor that gives rise to 28S18S and 5.8S runners. RNA polymerase II synthesize messenger RNA, many snRNAs, and several regulatory RNAs. RNA polymerase III synthesize transfer RNAs, 5S rRNA, and U6 snRNA. Among these, RNA polymerase II is the most important because it transcribes protein-coding genes. Promoters of RNA polymerase. Each RNA polymerase recognizes a distinct promoter architecture. RNA polymerase I promoters contain a core promoter and an upstream control element, or UCE. RNA polymerase II promoters may contain TATA Box, BRI, INR, DPE, MTE, CART Box, and GC Box. RNA Polymerase III promoters are unique because many of their promoter elements lie within the transcribed region itself and include Box A, Box B, and Box C. Thus, promoter organization varies according to the RNA polymerase involved. RNA Pol to core promoter elements. Let us examine the core promoter elements recognized during RNA polymerase II transcription. The BRI or TFI recognition element serves as the binding site for TFI. The TATA box is recognized by the TATA binding protein, or TBP. The initiator element, abbreviated INR, surrounds the transcription start site. The motif 10 element, or MTE, and the downstream promoter element, or DPE, contribute to promoter recognition, especially in TATA-less promoters. Different combinations of these elements create diverse promoter architectures and regulate transcription efficiency. General transcription factors. RNA polymerase II cannot initiate transcription independently. It requires a group of proteins called general transcription factors. These include TF2D, TF2A, TF2B, TF2F, TF2E, and TF2H. These factors assemble sequentially on the promoter before transcription begins. A useful mnemonic for remembering their order is DABFEH. These factors collectively recruit RNA polymerase II, stabilize promoter interactions, open DNA, and initiate RNA synthesis. Transcription factor 2D and TBP. TF2D is the first transcription factor to bind the promoter. It consists of two components: TBP, the TATA binding protein, and TAFs, which are TBP-associated factors. TBP recognizes and binds the TATA box. Binding of TBP induces a sharp bend in DNA of approximately 80 degrees. This DNA bending facilitates assembly of the remaining transcription machinery. TAFs recognize additional promoter elements such as INR and DPE. Pre-initiation complex formation. Assembly of the pre-initiation complex occurs in a highly ordered manner. First, TF2D binds the promoter. Next, TF2A stabilize this interaction. TF2B then binds the Br element. TF2F brings RNA Polymerase II to the promoter. TF2E subsequently recruits TF2H. The fully assembled complex is called the pre-initiation complex or PIC. This complex positions RNA polymerase II accurately at the transcription start site and prepares it for initiation. Transcription factor 2H and promoter melting. TF2H is one of the most important transcription factors because it possesses multiple enzymatic activities. Its helicase activity unwinds DNA and creates the transcription bubble. Its ATPase activity provides energy for promoter opening. Most importantly, its kinase activity phosphorylates the C-terminal domain or CTD of RNA polymerase II. This phosphorylation triggers promoter clearance and transition into elongation. Another important point is that TF2H also participates in nucleotide excision repair, linking transcription and DNA repair pathways. CTD of RNA Pol II. RNA polymerase II contains a unique structure known as the C-terminal domain or CTD. The CTD consists of multiple repeats of the heptapeptide sequence: tyrosine, serine, proline, threonine, serine, proline, serine. The CTD acts as a molecular platform that recruits proteins involved in transcription and RNA processing. Phosphorylation of specific serine residues determines the stage of transcription. Serine 5 phosphorylation is associated with initiation, whereas serine 2 phosphorylation is associated with productive elongation. Thus, CTD phosphorylation coordinates the entire TRAN. Elongation and PTEF. After initiation, RNA polymerase II enters elongation. An important elongation factor is PTEF, or positive transcription elongation factor B. PTEF phosphorylates serine 2 residues within the CTD. It also phosphorylates spt5 and recruits start SF1. These modifications convert RNA polymerase II into a highly processive elongation complex. Additional factors such as L and TFIIS assist elongation. TFIIS is particularly important because it helps RNA polymerase to recover from transcriptional arrest and improves transcription fidelity. Fact complex. Because eukaryotic DNA is wrapped around nucleosomes, RNA polymerase II encounters chromatin barriers during elongation. The fact complex, which stands for facilitates chromatin transcription, helps overcome this barrier. FACT consists primarily of spt16 and SSRP1 proteins. It temporarily removes H2A H2B histone dimers from nucleosomes, allowing RNA polymerase to pass. After polymerase passage, FACT helps reassemble nucleosomes. Therefore, FACT ensures efficient transcription while maintaining chromatin integrity. Pol I and Pol III transcription. Although RNA polymerase II receives most attention, polymerase I and III are equally important. RNA polymerase I transcribes the 45S precursor rRNA. Its transcription requires factors such as UBF and SL1. RNA polymerase III transcribes TRNAS, 5S rRNA, and U6 snRNA. Pol III promoters often contain internal elements such as Box A, Box B, and Box C. Specific transcription factors including TFIR, TFIEB, and TFIIC assist polymerase III transcription. These specialized transcription systems ensure proper synthesis of non-coding RNAs required for cellular function. Termination of eukaryotic transcription. Termination mechanisms differ among the three RNA polymerases. RNA Polymerase I termination requires specific termination factors. RNA Polymerase III terminates transcription at poly-U sequences, similar to bacterial intrinsic termination. RNA Polymerase II termination is explained by two major models. The first is the allosteric model, where recognition of the polydenylation signal causes conformational changes that destabilize the transcription complex. The second is the torpedo model. After RNA cleavage, the exonuclease XRN2, also known as RAT1 in yeast, degrades the remaining RNA attached to RNA polymerase II. When XRN2 catches the polymerase, transcription terminates. The AUAA sequence serves as the major polydenylation signal. Let us conclude with some high-yield points. RNA polymerase I synthesize large rRNAs. RNA polymerase II synthesize mRNA. RNA polymerase III synthesize tRNA and 5S rRNA. TBP is a universal transcription factor. TFIIH possesses helicase and kinase activities. CTD phosphorylation regulates transcription progression. FACT facilitates transcription through nucleosomes. Pol-3 promoters are internal promoters. TFIIR is essential for 5S rRNA transcription. The torpedo model involves XRN2-mediated termination. These concepts form the foundation for understanding RNA processing, which will be our next topic. Thank you. If you found this video helpful, please like, subscribe and share it with your friends.